All right, now that you all have had a little intro to a little bit of basic chemistry and macromolecules in our cards activity, we're going to get down to it and learn a lot more about these molecules and why they are so important. Okay, when we did our cards activity, you noticed that carbon came up all the time, right? We said it was the most ubiquitous. Well, why is that? What's the deal with that? And as it turns out, carbon is very unique. Out of all the elements in the periodic table, it is very special because it can form up to 4 single bonds. It can do double bonds. It can form super long chains. It can form super short chains. It can have things that branch off of it, and it can form rings. And no other element can do these things. Okay, not all of these things. And so because of all this variety of ways that carbon can bond, that's why we see it in so many molecules, and that's why we see it in the molecules that life is based on. Okay, so that's why it's carbon and not something else like oxygen or hydrogen or any of the rest of them, okay? And before we get any further, let's just be really clear about what a macromolecule is, okay? When we're talking about living things, as we learned, like organic means carbon containing, living things are organic. So living things are going to be made of molecules that are carbon based because of all of the bonding that can happen. And the molecules that make up living things are called macromolecules. Macro meaning like big, right? Opposite of micro, small, microscope, right? So these are not going to be teeny tiny little molecules like water. We're talking about big honking molecules that life forms, whether it's humans or plants or sea lions or whatever, they are all made of these same molecules, these macromolecules, okay? Just as a quick reminder, because we're going to be using these words a lot, we've got monomer and polymer. And the best analogy is a train. The polymer is the freight train and the monomer is the individual train car, right? You take a bunch of individual monomers and you bond them together through a covalent bond, and now you get a polymer. Okay, so now that we've got that out of the way, we're going to now dive into our 4 different types of macromolecules. Okay, we're going to be spending a couple slides on each one of these. This is not any special order, it's just the order that I happened to do them in. Okay, so first up are proteins. The monomer here would be an amino acid or a peptide. You can use either word, they're both equally correct. And the polymer would be a protein or a polypeptide. That just makes sense, because if you have one peptide, it's just that. But if you have many, poly means many, right? Polypeptide for the polymer. Get it? Maybe? I don't know. Okay. And this picture down here shows the chemical structure of an individual amino acid, so of one of the monomers. And all amino acids, there's a whole bunch of different ones, but all amino acids have the same basic structure. They have an amino group, that's this NH2 right here. And they have a carboxylic acid group, which is the C double bonded to an O with an OH. See where the name comes from? Amino carboxylic acid, amino acid, okay? And then there's going to be a carbon, at least one in the middle. But the main thing here is this R group, the side chain. And this is going to be different. Each amino acid is going to have a different side chain. It can look like lots of different things, as you saw on your cards. And that different side chain is going to give it different properties. So whether it works well with water or doesn't, whether it's polar or nonpolar, all these different things is going to be determined by the R group. Okay? Proteins are going to be super important. We're going to talk and talk and talk and talk about them all throughout the year. Most of your cells and like what they are and what they do, it's all determined by proteins. They do like all of the things. We have tens of thousands of different proteins. Okay, and each one has a specific 3D structure. And we're going to get more into this later, but for proteins, shape is really important. So shape is going to determine function, okay? A lot of proteins kind of work like a lock and key, and so if the lock and key don't fit together, they're not going to work, okay? So proteins, we will talk all about them. Here are just some examples. You don't need to write these down, but they do everything from like your immune system to hormones to like making up your muscles, all kinds of stuff. More on them all throughout the year. Lipids now, you know there's always that one special friend who just breaks the mold. And that's a lipid, okay? Because they don't really have a monomer or a polymer. But typically when we're talking about the structure of a lipid, we're going to say that there is a certain number of fatty acids. This is variable. And they're typically connected to a glycerol. This one shows 3 fatty acids, but it doesn't have 3. It could be 2. It could be a different number, okay? So here's that glycerol, which has 3 carbons, okay? And then here are these fatty acids, which are these just really long hydrocarbon chains. The main functions of lipids is going to be that they are going to basically be our membranes for things. Why? Because they're hydrophobic. So they are going to be water avoiding. So they're going to basically form little bubbles when they're in water. So this is going to form like our cell membrane. This is also going to form some of our organelle membranes. So that's going to be really important. Also energy storage. When your body wants to store energy, it stores it in a lipid, aka fat, right? And it's the highest density of energy storage your body can produce. And also things like cholesterol or the sex hormones like testosterone and estrogen, those are also lipids too. You might have heard of the term saturated and unsaturated. That has to do with the bonds. Notice saturated, they're all carbons. All the carbons has like 4 different things going on. Okay, 4 different single bonds. We got all these hydrogens in there, so it is saturated with hydrogens. Notice down here it's a different story. Oh, see here we've got this double bond. So now there's like 2 openings here. So it's unsaturated with hydrogens because like there's 2 open hydrogen spots. And as it turns out, that distinction is going to be really different because a saturated fat, because these are basically a line, they're going to be able to pack really closely together. And so they're going to be solid at room temperature. So this is found in like animal products, sausage, cheese, that kind of thing. Whereas an unsaturated fat, because of this double bond, it actually causes the molecule to bend. And so imagine if you put these all together, they've got a little bend in them, they're not going to pack quite as tightly together. And so this is going to mean that they're liquids at room temperature. This would be any of like the oils, they're plant-based things, so like vegetable oil, olive oil, all that good stuff. All right, next we've got nucleic acids. The monomer here is a nucleotide, and the polymer is going to be DNA, RNA, or also just a chromosome, okay? So what makes up a nucleotide? All nucleotides, whether they're DNA or RNA, have a similar structure. They all have a 5-carbon sugar. This could be deoxyribose in DNA, or it could be ribose in RNA. They all have a phosphate group. Here they're in circles. Okay, so they all have a phosphate group. And then coming off the other side of your 5-carbon sugar, there's going to be some kind of nitrogen base. That can be adenine, thymine, guanine, cytosine, or uracil if we're talking RNA. Okay, so those are our monomers. And then, of course, the function of nucleic acids is they're what allows us to pass on information to generation, right? Hereditary information that you inherit from your parents. And if you get down to the nitty-gritty, DNA's whole function is to give directions for making proteins, okay? So that is the main thing that our nucleic acids are going to do. Our last category are carbohydrates. The monomer here is a monosaccharide, or it's also called a simple sugar. The polymer is a polysaccharide, or you just hear the word carbohydrate, or just a sugar. And the chemical structure can vary, but typically you've got carbon chains or rings, more on that in a second. And your carbons, most of your carbons have an OH bonded to them somewhere. That's called a hydroxyl group. And one carbon is going to have a double bond to an O somewhere, and that's called a carbonyl group. Okay, and just the different sugars have different structures. Like I said, they could often be in a ring shape. So here's glucose in a line, glucose in a ring. You don't need to worry about this, but just know that they can exist in either form depending on what environment they're in. And carbohydrates, their main function is for energy storage, right? You get energy from sugar. We all know this. And so our cell is going to use this as its main form of nutrient. Glucose is going to be your cell's main energy source. If you see something that ends in OSE, that's usually a carb, right? Glucose, fructose, galactose, all carbohydrates. In addition to being energy storage and giving us nutrients, it also provides structures. Things like cell walls, exoskeletons are primarily carbohydrate based. One last thing here would just be to really emphasize that it doesn't matter which macromolecule we're talking about. All atoms are held together by covalent bonds. So between this N and this H, covalent bond. Between this N and this C, covalent bond. Same thing. All of these bonds that are symbolized by 1 or 2 lines for double bond, they're all covalent. Same thing here. Every single bond you're seeing is covalent. Even over here, all of these crazy bonds and these shapes, they're all covalent. So they are all sharing electrons, okay? No ionic, no hydrogen with this. This is all covalent bonding here. If you want to know more, this is chapter 3 in the book, and I highly recommend you go and you look through these pages in the book. Now that you've had a lecture on it, you should be able to read through it. This should be mostly review. You can see more pictures, hear more examples, get a better feel for things. And then here are the main things that this lecture covered, so that later when you're maybe preparing for a quiz or a test and you want to make sure you've got the main important ideas, you can go through this list and quiz yourself and see how you're doing. As always, if you have questions, make sure you write them down. You'll get a chance to answer them, to ask them in class. So please, if anything didn't make sense, make sure to ask it in class when we take that time.